Transcription and processing of RNA. In this chapter, we are going to learn about transcription, site and time of occurrence, sense and antisense strands, mechanism of RNA transcription, materials required, RNA polymerase in prokaryotes, transcription unit and transcription promoter site, functional or consensus sequences and promoter region, molecular mechanism of transcription in prokaryotic cells, molecular mechanism of transcription in eukaryotic cells, eukaryotic polymerases, functional sequences in promoter region in eukaryotes, RNA-directed RNA synthesis, then post-transcriptional modifications of RNA or RNA processing, then processing of tRNA and rRNA, then processing of mRNA, spliceosomes, RNA synthesis in vitro, comparison between replication and transcription. Transcription. DNA contains information for the synthesis of cell-specific proteins. DNA is located in the nucleoid in prokaryotes and nucleus in eukaryotes, and protein synthesis occurs in the cytoplasm. DNA does not move to the site of protein synthesis, ribosomes, to directly guide the process. Instead, it transfers its information to single-stranded mRNA molecules which move to the ribosome to direct protein synthesis. The process of the formation of all the three families of RNA, mRNA, tRNA, and rRNA from DNA template is called transcription, meaning written across. It involves rewriting the genetic message coded in DNA into RNA molecule. The transcribed mRNA moves out of the nucleus to the ribosomes in the cytoplasm and directs protein synthesis. Site and time of occurrence. Transcription occurs in the nucleus during the G1 and G2 phases of interphase. Then DNA synthesis does not occur when the cell is dividing. DNA has promoter and terminator sites. Transcription starts at the initiation point close to the promoter site and stops at the termination site. Enzyme polymerase recognizes a promoter sequence of the gene and binds to its initiate transcription. During transcription, only one of the two strands of DNA duplex is transcribed. As in DNA enzymes, polymerase can add nucleotides to the 3' OH end of the polynucleotide chain. RNA chain is also synthesized in 5' to 3' direction. It is transcribed in 3' to 5' DNA strand. The 3' to 5' DNA strand from where RNA is transcribed is called template strand or antisense strand or non-coding strand. Its complementary strand or 5' to 3' strand is called anti-template strand or sense strand or coding strand, because it has the same sequence of nitrogenous bases as the RNA transcript, except T of coding strand is substituted for U, and it also has the same polarity. Then extension, sense and antisense strands. The transcribed RNA has 5' to 3' polarity and it is called sense RNA. It is transcribed in 5' to 3' direction on the 3' to 5' strand of DNA, which is called a template strand, because it acts as a template for RNA synthesis. 3' to 5' strand or template strand is also called antisense strand because its polarity is just opposite to RNA strand and its nucleotide sequence is complementary to sense strand. 3' to 5' strand or template strand is also called antisense strand because its polarity is just opposite to RNA strand and its nucleotide sequence is complementary to sense RNA. The 5' to 3' strand or complementary strand of DNA is called sense strand because the sequence of nitrogenous bases in this strand is similar to that of RNA, except T is substituted by U. Since RNA is the coding instrument for protein, the non-template or sense strand is also called coding strand and template strand as non-coding or anti-coding strand. Mechanism of RNA transcription in prokaryotes. The mechanism of RNA synthesis was worked out in late 1950s by American investigators Gerald Horowitz and Samuel B. Swiss and Audrey Stevens in vitro experiments. The existence of mRNA was first shown by Spiegelman and co-workers, and RNA polymerase activity was discovered by Wiss in 1960. So RNA transcription requires the Enzyme RNA polymerase or DNA-directed RNA polymerase, DNA template, the transcription unit, all the 4 types of ribonucleoside triphosphates ATP, CTP, GTP and UTP, divalent metal ions Mg2 plus or Mn2 plus as a cofactor. No primer is needed for RNA synthesis. Core enzyme sigma factor RNA polymerase enzyme of Bacterium E. coli is showing association to its polypeptide. So RNA polymerase in prokaryotes. Bacterial RNA polymerase is a complex holoenzyme that catalyzes transcription of RNA. In prokaryotes, single RNA polymerase RNAP enzyme carries out synthesis of all the 3 types of RNA. But in eukaryotes, 3 distinct RNA polymerase enzymes, namely RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3 catalyze synthesis of rRNA, mRNA and tRNA respectively. RNA polymerase 2 and RNA polymerase 3 catalyze synthesis of rRNA, mRNA and tRNA respectively. In addition, mitochondria and chloroplasts have their own RNA polymerases which resemble bacterial enzyme. In prokaryotic, single RNA polymerase catalyzes synthesis of all the 3 types of RNA. In E. coli, RNA polymerase enzyme is a large protein with a molecular weight of 480,000. It is a holoenzyme and consists of core enzyme and sigma subunit. Core enzyme is consists of core enzyme is there, then sigma factor is also there. Sigma subunit and core enzyme. Just write that down. Core enzyme and a sigma factor. Core enzyme and a sigma factor. Okay. So core enzyme is formed of 4 tightly associated protein chains, namely different kinds with molecular weights associated with that. Then it binds non-specifically to DNA. Template and migrate downstream till its sigma factor recognizes the promoter region. And site for binding to sense strand of DNA, enzyme movement, active sites on bacterial core enzyme of RNA polymerase. Active site on bacterial core enzyme. Active site on bacterial core enzyme of RNA polymerase. RNA binding site, RNA DNA hybrid, site for binding to multi antisense or template strand of DNA unbinding site, coding of sense strand of DNA, then DNA unwinding pre-binding point and RNA. These are the points. Then extension functional site on core enzyme of RNA polymerase. The core enzyme has following 4 functional sites. DNA unwinding site unwinds DNA duplex and separates 2 nucleotide strands. The enzyme moves along DNA duplex to be transcribed. Site binding to antisense strand or template strand of DNA molecule. Site binding to the sense strand or coding strand of DNA molecule. DNA rewinding site binds to site where separated sense and antisense strands of DNA start rewinding after the transcription of that segment of DNA is complete. The sigma subunit. The core enzyme has 2 molecules of sigma polypeptide. This is coded gene RPOA. The alpha subunits are needed for the assembly of core enzyme and probably help in the recognition of promoter. The beta promoter, only one copy of beta subunit is present. In one molecule of core enzyme of RNA polymerase, it is encoded by gene RPO beta. The beta subunit binds with the incoming nucleotide to be added to RNA chain and helps in the formation of the first phosphodiester bond. 2 beta subunits, it is encoded in the RPO-C gene and binds with the template strand or antisense strand of DNA. Therefore, both beta and beta minus catalytic subunit of core enzyme and sigma subunit. Sigma subunit is formed of a single polypeptide gene loosely attached to the core enzyme. It is encoded by the gene RPOD. It recognizes the start signal of the DNA molecule and directs the core enzyme of RNA polymerase to bind to the promoter region upstream of initiation codon. The sigma subunit recognizes two special sequences of bases in the promoter region and coding strand, non-template strand of DNA. There are 10 minus 10 sequences and minus 35 sequences. Therefore, ability of holoenzyme RNA polymerase to recognize promoter sequence is due to sigma subunit, which also facilitate opening or melting of DNA helix. The sigma subunit separates from core enzyme once about 10 nucleotides are joined to initiate RNA transcription. The core enzyme without sigma factor binds non-specifically to any region of DNA and can transcribe DNA there because it fails to recognize specific promoters on DNA template. It is also unable to discriminate between the two strands of DNA helix to be used as a template. Sigma factor recognizes specific transcription initiation sites where RNA synthesis can begin. The sites are called promoter sites, and these are different sigma factors, one for every promoter site. Then functions of RNA polymerase. RNA polymerase unwinds about 15 bases of DNA around the initiation site to form an open promoter DNA complex and provides a single strand of DNA to act as template for transcription. RNA polymerase basically catalyzes the formation of phosphodiester bonds between successive nucleotides of a polynucleotide chain during synthesis of both DNA and RNA. With sigma factor, it continues to synthesize RNA because sigma factor recognizes promoter site. RNA polymerase lacks proofreading 3 to 5 exonuclease activity. Therefore, one error for every 10 raised to 4 to 10 raised to 5 ribonucleotides incorporated is introduced during RNA transcription. But the mistake during RNA transcription is not serious because of its high turnover and opal pairing during translation. So the transcription unit is a stretch of nucleotides in DNA that is transcribed into a single functional mRNA molecule. It is called an operon in prokaryotes and a gene in eukaryotes. A typical transcription unit has following essential parts. Which are they? A promoter region, a start point or initiation sequence, a coding segment, a terminator sequence. RNA polymerase binds to the promoter. Transcription begins at the start point, progresses along the length of coding sequence, and terminates at the terminator sequence or at the termination codon. The transcription of mRNA starts in 5' to 3' direction on 3' to 5' strand of DNA. Promoter site or promoter region and functional or consensus sequence in promoter region in prokaryotes. Promoter site is a segment of regulatory DNA located just preceding the initiation codon of a cistron or gene beyond the 5' end of coding DNA. Enzyme RNA polymerase binds to this site for the initiation of transcription. This stretch of DNA in front of structured gene is also called upstream region. And the sequence of bases in this region is referred to as upstream sequence. It is highly variable site having specific sequence of 20 to 200 base pairs. In E. coli, it consists of about 70 base pairs located before the transcription start site. The promoter region in prokaryotes has 2 to 3 important functional sequences. These are called consensus sequence. The sequences from different systems or from promoters of different cistrons differ only by 1 to 2 bases. These functional sequences are 10 sequence or Pribnow box, minus 10 sequence or a Pribnow box. It is a consensus sequence of 6 to 10 bases upstream to the initiation site of mRNA. In E. coli, it occupies 10 regions of promoter. All sequences in Pribnow boxes of different genes are variants of 5' TATA80 3'. In this segment, at the position of 6T, it's present in its promoter and is called conserved T. The sequence of bases in 10 region of promoter is named Pribnow box after its discoverer, David Pribnow. So Pribnow box requires sequences orients DNA polymerases on promoter and helps it in locating the precise base at which transcription will begin. So consensus and conserved sequences, two types. A consensus sequence is the base sequence in a DNA strand that is present in all promoters or in other regions of DNA which perform the same function but with some variation. It means consensus sequences from different sources are similar in structure and function but not identical. They vary from one group of organism to another. In contrast, a conserved sequence remains unchanged in all DNA sequences having the same function. Upstream region, spacer coding region, then spacer consensus sequence, initiation codon, direction of transcription. Base sequence of promoter site encoding or non-template DNA at E. coli, a different region in the promoter site, consensus sequence in promoter region of tryptophan gene, promoter region of lac gene, promoter DNA, TATA box sequence, start codon, etc. Transcription DNA, coding strand, template strands. A promoter region in prokaryotes E. coli, a base sequence of promoter site in DNA of E. coli. The key regulatory sequences for initiation of transcription are found in this region, located at 35 and -10 base pairs upstream from the initiation site. Then consensus sequence in -35 region. The consensus sequence in -35 region occurs as a variation of 5' TTGA 3' prime. It lies about 35 bases upstream from the initiation site. Because of its location, this consensus sequence is called the 335 sequence. This hexanucleated sequence is recognized by sigma factor of RNA polymerase. The sigma subunit of RNA polymerase binds to -35 sequence. Owing to the great size of the enzyme, the appropriate region of polymerase corners to lie in contrast with Pribnow box and start unzipping of DNA. Thus, there are different sigma factors to recognize and attach to different versions of hexanucleated sequence. Upstream promoter element. There is another AT recognition element in the promoter region that is present only in the promoter of certain highly expressed genes, and it is located between 40 and 60 base sequences. This upstream promoter sequence binds to the alpha subunit of RNA polymerase. Molecular mechanism of transcription in prokaryotic cells. The molecular mechanism of transcription involves the following steps: elongation of promoter region on DNA. The sigma factor of RNA polymerase recognizes 35 region and 10 region of promoter. The two enzymes of RNA polymerase then binds tightly to the promoter region. So loose binding, holoenzyme, DNA polymerase scans double helix. Promoter region of DNA polymerase binds tightly to promoter region. Core enzyme polymerase unwinds DNA. Transcription begins. Recognition of promoter region on DNA by DNA polymerase and unwinding of DNA. So binding of RNA polymerase to DNA duplex. The core enzyme RNA polymerase binds to a specific promoter in the DNA double helix. The site is located on the 5' end upstream of the initiation site of the gene to be transcribed. It initiates separation of DNA strands and thus determines where to start DNA synthesis. The promoter also determines which DNA strand is to be transcribed. Promoter, structural gene DNA, nonsense strand, sigma subunit, core enzyme inactive, then pre-transcriptional DNA sigma factor, DNA polymerase, replication bubble, sense strand or master strand, template binding, then initiation, elongation, termination, and completion. These are the steps, different steps of transcription. Exposure of DNA bases and initiation of polynucleotide gene. RNA polymerase initiates local unwinding and separation of two strands of DNA. The separation begins from about the middle of the Pribnow box, which is 6 to 10 base long segment of DNA prior to the initiation point of RNA synthesis. Approximately 10 to 15 base pairs are separated, leading to the formation of transcription bubble. So there is a structure called a transcription bubble. So the separation of DNA strands exposes the basis of the DNA for the initiation of synthesis of polynucleated chain of mRNA. After the two DNA strands, only one 3' to 5' strand functions as template. It is called a master strand or a nonsense strand or template strand. The complementary strand corresponding to master strand is called coding strand, and sense strand or non-template strand. Master strand is there, and along with that a coding strand is there. Coding strand, master and coding. Okay, then base pair. The ribonucleotide triphosphate molecules, adenosine triphosphate, guanosine triphosphate, and cytidine triphosphate (CTP) and uridine triphosphate (UTP) start binding to the nucleotides of antisense strand or template strand of DNA from the initiation point. The ribonuclear proteins are found floating in the nucleus and serve as a raw material for RNA synthesis. These are found by the activation phosphorylation of ribonucleoside monophosphates. Adenosine monophosphate AMP, guanosine monophosphate GMP, cysteine monophosphate CMP, and uridine monophosphate through union with ATP. The enzyme phosphorylase catalyzes the activation process. The ribonucleoside triphosphate get joined to the base of the DNA template chain one by hydrogen bonding according to the base pairing rule, and it's thought about by RNA polymerase. This enzyme RNA polymerase not only initiates but also catalyzes polymerization. Elongation of ribonucleotide chain is 5' to 3' direction. An extension. The first transcribed base for mRNA is normally A, but sometimes it may be G. However, it is never a pyrimidine. About 40 nucleotides per second are added to the growing RNA chain. This is how slower the DNA duplicates, in which about 1000 nucleotides are added per second. Then conversion of ribonucleoside triphosphate to ribonucleoside monophosphate. The various ribonucleoside triphosphates on linking to the DNA template chain break off near high energy bonds. This changes them to ribonucleoside monophosphates that are the normal components of RNA and sets free pyrophosphate group. Pyrophosphate contains a high energy bond. It undergoes hydrolysis by the enzyme pyrophosphates. This releases energy and sets free inorganic phosphate. The first ribonucleotide triphosphate retains all the three phosphatases and is chemically distinct from other nucleotides added after it. P plus H2O pyrophosphatase 2PI plus energy. Elongation of RNA polynucleotide chain. With energy so released, each ribonucleotide monophosphate joined to DNA template chain, then join the ribonucleotide arrived earlier, making the RNA chain longer. The enzyme RNA polymerase catalyzes the formation of phosphodiester bonds between successive nucleotides and require an divalent ion Mg++ and Mn2+. When a few nucleotides, about 10, are added, the sigma factor is released from the core of the enzyme RNA polymerase. About 10 to 12 nucleotides of the growing DNA chain are bonded to template DNA at any one time. As the polynucleotide chain grows beyond 12 nucleotides, it begins peeling off DNA template. mRNA synthesis on DNA template take place in 5' to 3' direction, so that the successive nucleotides are attached at the 3' end of growing RNA strand. Core enzyme moves along DNA and unwinds the two strands, exposing the nucleotides of template DNA. As a result, complementary copy of RNA is transcribed on 3' to 5' strand of DNA. The transcribed RNA molecule is called RNA transcript or primary RNA transcript. Then transmission of transcription and separation of RNA chain. As transcription proceeds, the hybrid DNA RNA molecule dissociates, partly freeing the RNA molecule under synthesis. When the polymerase core enzyme reaches a terminator sequence on this DNA, it leaves the DNA. The terminator sequence lies in the template strand and consists of two inverted repeats starting by about 6 bases and followed by a sequence of A's. The starting of A's in template gave rise to a sequence of U's in the 3' end of mRNA. It is the terminator sequence in mRNA. The inverted repeats in mRNA are complementary and can pair to generate a hairpin structure. A long RNA molecule can have many hairpin structures. They provide an opportunity of termination, but termination occurs at any position within the sequence of U's. The fully formed RNA chain is now forms several molecules of RNA which are released one after the other on completion from the RNA template. Equally, two methods of termination have been identified: without termination factor and with the termination factor, row dependent and row independent. First, row protein dependent termination dependent. The chain termination process, row, acts as an accessory termination factor. It acts as a disrupting the RNA DNA hybrid. It dissociates the DNA transcript from template and terminates transcription. Row protein is a specialized helicase. It consists of a hexamer of 6 identical subunits consistent bound to the sequence of 50 to 90 base located upstream of the row binding sequence is rich in C and poor in G. Row binds really into growing mRNA. Pro-protein independent termination. This type of RNA transcript termination depends on a specific DNA molecule that ends RNA transcript with a stable hairpin structure. Such termination sites have two or more stretches of G-C pairs arranged in the form of inverted repeat, followed by a string of adenine poly A in the master strand. The released mRNA is called the primary transcript. The self-complementary residues in the transcript fold into a hairpin loop, ending a poly U sequence at 3' end. It undergoes processing to become functional. From nucleus, it comes out into cytoplasm through nuclear pores. Termination of RNA transcript from DNA template without the Rho factor in prokaryotes. The termination region contains two stretches of G to C pairs arranged in the form of inverted repeats. And reformation of DNA helix. Difference between replication and transcription. The differences in replication and transcription are the replication new DNA molecule are formed in the transcription RNA molecule is synthesized. Uracil base is introduced into DNA in the thymine base. RNA transcripts do not remain connected and not separate out as single strand is out of the nucleus of the cytoplasm and there is synthesis of protein. Several copies of RNA transcripts are released into each DNA template. Then reformation of DNA helix. As the DNA chain grows, the transcribed region of DNA molecule gets hydrogen bonded to the sense strand and the two are spirally coiled to assume the original double helical form. When the last nucleotide is added, the RNA polymerase and RNA chain are fully released from the DNA, which also completes winding into the double helix. The protective protein coat is added again to the DNA helix or duplex. Processing of RNA. The originally transcribed RNA molecule is called primary transcript. In both the prokaryotes and eukaryotes, they undergo extensive changes to become functional. This is called processing of rRNA or RNAs or post-transcriptional modification of RNAs. Molecular mechanisms of transcription in eukaryotic cell. Transcription in eukaryotic cells is more complex and differs from that in prokaryotes in following features. 3 different RNA polymerases are required in the transcription of 3 different types of RNAs. Eukaryotic promoters are more varied than prokaryotic promoters. There are 3 different types of promoters for 3 different polymerases. Some eukaryotic promoters are located downstream from the transcription start point, while some are present upstream. So, differences between prokaryotic and eukaryotic transcription. ഒന്നാമത് prokaryotic transcription occurs in the cytoplasm, but eukaryotic transcription occurs in the nucleus. Prokaryotic transcription, there is no definite phase for its occurrence, but eukaryotic transcription takes place in the G1 and G2 phases of cell cycle. Then prokaryotic transcription, a single RNA polymerase synthesizes all the 3 types of RNA, mRNA, tRNA and rRNA. Coupled transcription translation in the role. Then initiation of transcription does not need any protein or initiation factors. Pre-RNA molecules are released and processed in the cytoplasm. RNA polymerase is a complex set. 5 polypeptides. Transcriptional unit has more than 1 genes. Polycystonic. The mRNA primary transcript has fewer surplus nucleotides. But eukaryotic transcription, 3 RNA polymerase 1, 2 and 3 synthesize rRNA, mRNA and tRNA respectively. Coupled transcription translation is not possible. Initiation of transcription require proteins called transcription factors. These are TF2A, TF2B, TF2D and TF2E, TF2F and TF2H. This recognized data box. So pre-RNAs are released and processed in the nucleus. RNA polymerases are complexes of 10 or 15 polypeptides. Transcriptional unit has only 1 gene. Monocystonic. The mRNA primary transcript has a large number of surplus nucleotides. Transcription factors. Many transcription factors are involved in binding of eukaryotic RNA polymerase to DNA. Unlike prokaryotic sigma factors, the transcription factors are not part of RNA polymerase and bind to promoters even before the polymerase binds to initiate a transcription. So upstream control element is there, core promoter, start point and transcription will be taking place. That is the promoter of RNA polymerase 1. Which are the regions? Upstream control element, DNA, core promoter, start point and transcription. Then core promoter for RNA polymerase 2, TATA box or TATA box, initiation, then start point, template DNA. These are some major regions and transcription is also taking place. And promoter for RNA polymerase 3, promoter for tRNA gene, start point, box 1, box A, box B, transcription. Then promoter for rRNA gene, start point, box C, transcription. Different promoters for 3 different types of RNA polymerase 1, 2 and 3 in eukaryotes. Promoter for mRNA is upstream, but for tRNA and rRNA is downstream of start point. The 3 nuclear RNA polymerases are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So RNA polymerase 1, it is found in the nucleolus and is responsible for the synthesis of ribosomal RNA. The RNA molecule synthesized in the nucleolus is precursor to 3 ribosomal RNAs. Then it is found in the nucleoplasm and transcribes protein coding gene. The synthesis precursors of mRNA. It also synthesizes most small nuclear RNAs which are needed for post-transcriptional processing of primer RNA. And RNA polymerase 3. It is also found in nucleoplasm. It is responsible for the synthesis of a variety of small RNAs including tRNA and 5S rRNA. The 3 eukaryotic polymerase enzymes are large and formed of many polypeptide subunits. Their molecular weights is about 5 lakh. For example, eukaryotic RNA polymerase 2 has more than 10 subunits and 3 of them are also present in RNA polymerase 1 and 3. The largest subunit for RNA polymerase 2 is related to beta subunit of prokaryotes. Okay, there are specific proteins present in the nucleoplasm and are required for the correct functioning of RNA polymerases during initiation of RNA transcription. They bind to TATA box and are also called TATA binding proteins. These are classified into 2 subgroups like general transcription factors or basal transcription factors. These are needed for the transcription of all the genes transcribed by a particular RNA polymerase. They are designated as TF1, TF2 and TF3 and correspond to the 3 RNA polymerases. Promoter of RNA polymerase 2 is their initiation site gene, initiation box, then TATA box and TF2D T-gen start point. TF2D recognized promoter for RNA polymerase 2. TF2A and TF2B also bind a promoter. Then DNA RNA polymerase 2 and TF2F bind to form pre-initiation complex. Then binding of TE2E, TF3H, TF2J to pro-initiation complex. Then TF2H phosphorylates the tail of RNA pol 2 and other factors leaves and RNA polymerase 2 moves forward along DNA. So formation of pre-initiation complex during initiation of transcription in eukaryotes, steps in the formation of transcription apparatus and movement of RNA polymerase 2 forward from the binding site. Additional transcription factors or specific transcription factors. These are needed for the transcription of particular specific genes under specific circumstances. These are responsible for regulating gene expression and therefore control the expression of individual genes. Transcription factors needed for RNA polymerase 2. Transcription factors TF2B, TF2D, TF2E, TF2F and TF2H are general, which are the B, D, E, F and H are the general transcription factors required for RNA polymerase 2 to start RNA transcription. TF2D has a TATA binding protein which binds specifically to the TATA consensus sequence. TF2D also binds to the TBF2FB forming Tata TF2B complex at the promoter. With the help of TF2F factor, RNA polymerase binds to its complex. The release of RNA polymerase 2 from promoter and its movement along template DNA require other the TF2 complexes. TF2E, TF2H and TF2U, TF2H unwinds DNA around the transcription site. Helicase activity and phosphorylates the tail of largest subunit of RNA polymerase at the C terminal. This end is also called the carboxy terminal domain. It consists of 7 amino acids repeated approximately 50 times. On being phosphorylated at serine or threonine residues, polymerase is released from promoter region to slide over the template strand of DNA. All other TF2 complexes except for TF2H are left behind. The RNA polymerase moves forward. Transcription factors associated with RNA polymerase 2 binds to data box. A part of TF2D includes TBF, recognized polymerase 2 specific promoter. First to bind to data box, binds upstream of data box required for binding of RNA polymerase 2 to promoter, binds downstream of data box required for binding of RNA polymerase 2 to promoter, accompanies RNA polymerase 2 as a binding of promoter. These are the functions performed by TBF, TB2D, TE2A, etc. And which are the binds to data box, part of TF2D, which one TBP. Then next include TBP like that. Then functional or consensus sequence in promoter regions in eukaryotes. Sequences of nitrogenous bases similar to that found in prokaryotes are present in the promoter region of eukaryotes also. There are different promoters for all the RNA polymerase. Promoters for polymerase 2. The promoter region for RNA polymerase 2 is found in 3 regions: the initiator box, data box, and a variety of upstream elements. Data box or Goldberg-Hagen's box. Goldberg-Hagen's box. Okay. So the consensus sequence is data box is a hexanucleotide sequence consisting of 5' TATAAA 3'. It is usually represented by TATXAX, where X and X may be T or A. It is comparable to Tribnov sequence of prokaryotic promoter. It is the site of assembly of a pre-initiation complex and determines the site of initiation of transcription. It lies about 25 BP upstream of initiation box. It is rich in ET sequence. The assembly of the initiation complex, transcription factor, and polymerase is essential for initiation of transcription. In addition, eukaryotic genome has proximal control elements named GC box with base sequences. Then promoter for RNA polymerase 1. The promoter of RNA polymerase 1 has an upstream control element and a core promoter. The core promoter site is rich in GC sequence. The protein UBFI upstream binding factor recognize the binds to both the upstream control element and the core promoter later. SL1 selective factor for pol 1 binds to DNA association with UBF1. Usually RNA polymerase 1 binds to transcribed rRNA genes. Promoter for RNA polymerase 3. The promoters of RNA polymerase 3 for both 5S rRNA, RNA and tRNA are located within the gene itself. The recognition sites for them are downstream for the start site. Two factors TF3C and TF3B are needed for the binding. Firstly, TF3C for TF3A binds to both sites and induces TF2B and bind to the promoter near the start site, and TF2B binds RNA polymerase 3 to promoters in DNA. An RNA-directed RNA synthesis. The RNA viruses whose genome is RNA is transcribed from RNA. This viruses induces the formation of an enzyme RNA-directed RNA polymerase or RNA replicase in the host cell. RNA replicase acts like DNA-directed RNA polymerase and exhibits the following characteristics. RNA replicates directs the synthesis of RNA on RNA in 5' 3' direction. Ribonucleotides are added to the RNA end in the same fashion as in DNA directed RNA polymerase. Ribonucleotide triphosphate either in the chain on 3' end and pyrophosphate is released. The RNA transcript is complementary to the RNA template. Editing, proofreading, and repair are not associated with RNA replicase. Then RNA replicase works on single stranded template RNA. Next, RNA editing. RNA editing is the post-transcriptional modification of primary mRNA transcript by the action of a deletion of or both addition and deletion of one or more nucleotide in pre-mRNA. This phenomenon was observed in mild 1980s in the production of certain mRNAs in mitochondria of some protozoa. The conserved protein subunit 3 of cytochrome oxidase is encoded by mtDNA. The sequences of COH3 gene and its mRNA transcripts were transcribed in 3 protozoans, Trypanosoma brucei and some other substances. It was found that the mtDNA sequence of this gene by CF and L1 were collinear with the mRNA but not in TB. The mRNA and TB had several U nucleotides inserted which were not encoded in the DNA. The DNA of TB had several T nucleotides which were not represented in the transcript. There is extensive insertion of U nucleotides in mRNA and deletions of several T nucleotides. This editing is carried out by a special RNA molecule called guide DNA or gRNA. It pairs with mRNA transcript and causes the cleavage of the transcript, templating the missing U nucleotides and ligating the transcript. Editing may involve addition of single C nucleotides at many positions or substitutions of C to U or U to C. The C to U editing is found to result in the formation of an A to U AU initiation codon from AUG to join the some chloroplast mRNAs in some higher plants. It is assumed that RNA editing process is an additional regulatory control process and results in the generation of tissue-specific initiator or stop codon. C to U editing is found in the mRNA transcribed from a nuclear gene that encodes for cytoplasmic apolipoprotein B to mammals. So, post-transcriptional modification of RNA or RNA processing. The immediate product of transcription is called a primary transcript. It is a biologically inactive. The process of chemical modification in the primary transcript to generate the final functional RNA product is called RNA processing. The RNA processing, largest RNA precursors are cut into smaller RNA by ribonuclease P enzyme. Cleavage. Unwanted nucleotides are removed by enzyme named nucleases. Splicing. Usual regions are rejoined by ligase enzyme. Then union. Certain nucleotides are added at the end of enzymatic terminal region or terminal addition. Molecules may fold on itself to assume proper shape or folding. The immediate product of transcription is called primary transcript. It is biologically inactive. The process of chemical modifications in the primary transcript to generate the final functional RNA product is called RNA processing. In RNA processing, larger RNA precursors are cut into smaller RNAs by ribonuclease P enzyme. Cleave. Unwanted nucleotides are removed by enzymes named nuclease. Splicing. Unusual regions are rejoined by ligase enzyme. Union. Certain nucleotides are added at the end enzymatic terminal addition. A molecule may fold on itself to assume proper shape, folding, and some nucleotides may be modified. Nucleotide modification. Primary transcript and the ribonuclease P enzyme. Primary transcript is RNA molecule formed immediately after transcription. The primary transcript of 3 different types of RNAs before undergoing processing are known as precursors or pre-mRNA, pre-mRNA and pre-tRNA and pre-rRNA. In eukaryotes, pre-mRNA are also referred to as hnRNA or heterogeneous nuclear RNA. So, ribonuclease P enzyme, the enzyme ribonuclease P is a riboenzyme. It is formed of both a protein and a small RNA molecule. The catalytic activity of enzyme is due to RNA present in the large. Processing of pre-RNA transcript. Before that, post-translational modification. Primary transcript is RNA molecule formed immediately after transcription. The primary transcripts of three different types of tRNA, rRNAs. RNAs before undergoing processing are known as precursors or pre-mRNA, pre-tRNA and pre-rRNA. In eukaryotes, pre-mRNA was also referred to as hnRNA or heterogeneous nuclear RNA. Then ribonuclease P enzyme. The enzyme ribonuclease P is a ribozyme. It is formed of both a protein and a small RNA molecule. The catalytic activity of enzyme is due to RNA present in the large subunit ribosome. The protein component merely modulates the activity of RNA component. So processing of pre-tRNA transcript. Eukaryotic pre-tRNA molecules are biochemically altered. The process has been studied in the yeast, where following four processes are needed for functional tRNA molecule to form: trimming of ends by the cleavage of phosphodiester bonds, splicing or removing an intron, an addition of some terminal sequences, heterocyclic base modification used by methylation. In prokaryotes, pre-tRNA molecule undergo only a few of the above modification steps. In some prokaryotic tRNA, some introns are removed, in some terminal sequences are added, and in some others, a few heterocyclic and ribose rings are modified. The endonuclease enzyme ribonuclease P catalyzes removal of a segment from 5' end of the pre-tRNA by hydrolytic cleavage. And processing of rRNA transcript. The primary transcript of rRNA contains precursors for all the three rRNAs. The processing involves the trimming so smaller segments of appropriate size with the help of ribonuclease. It has already been discussed in biogenetics of rRNA. Then processing of pre-mRNA transcript. Most prokaryotic mRNA transcript require very little or no post-transcriptional modification to be changed into functional mRNA. In prokaryotes, often translations of mRNA begins while it is being transcribed. This is called coupled transcription translation. But in eukaryotes, the primary mRNA transcript molecules have copy of both exons and introns. Therefore, they are spliced and chemically modified in the nucleus before being transported to the ribosome for translation. Following three biochemical processes occur during mRNA processing in eukaryotes: RNA capping at 5' end, addition of polyadenylate tail at 3' end, as splicing or removing of introns. RNA capping. The 5' end of primary mRNA transcript is modified by the removal of a phosphate from the triphosphate functional group. A guanine monophosphate gets attached to its diphosphate at 5' end, resulting in the formation of an unusual 5' triphosphate covalent linkage. The guanine residue at the end of this linkage is methylated at N2. The methylated guanosine forms a cap at 5' end of the mRNA. Additional capping may occur at another methylation of hydroxyl groups in ribose. The first one or two nucleotides and still higher from higher eukaryotes. Only cap O is formed lower eukaryotes. Poly A addition. The 3' end of polymerase mRNA transcript is modified by the addition of a polyadenylate, poly A tail. It is catalyzed by polyadenylate polymerase, poly A polymerase enzyme. The poly A tail is formed of 20 to 250 nucleotide residues. Before the addition of poly A tail, an endonuclease removes the few nucleotides from the 3' end of the transcript. The cleavage site has still recognition sequence, and it is recognized by polyadenylation complex. The polyadenylation complex is formed by the joining of poly A adenylate and poly A binding protein with cleavage polyadenylation specificity factor and cleavage stimulation factor. This complex binds to the tail recognition sequence and GU-rich sites and stimulates the process of cleavage of nucleotides and addition of poly A tail. Most eukaryotic mRNA molecules have a 3' poly A tail. This is thought to stabilize mRNA molecules by increasing resistance to cellular nuclease and helps it to cross the nuclear membrane. RNA splicing. The process of cutting out introns from the primary mRNA transcript and joining the exons to form functional mRNA is called RNA splicing. It occurs in the nucleus of only eukaryotic cells which have split genes. Mechanism of RNA splicing. The mechanism of RNA splicing depends on specific base sequences at the junction between introns and exons. The consensus sequence at the 5' end of mRNA intron called the 5' splice site starts with the consensus sequence 5' GU and 3' splice site at 3' end of the intron ends with consensus sequence 3' EG. In the middle of intron is present a special adenine residue. It is used as a branch site during splicing. The consensus recognition sequence for the sites are 5' splice site, 3' splice site and branch site. Spliceosome or splicing apparatus. The 5' and 3' splice sites and the branch sequence are recognized by a complex formed of proteins and small segments of RNA. This RNA protein duplexed together from a large splicing apparatus called spliceosome. This complex brings 5' and 3' splice sites in intron together. Spliceosome is a phage body formed of snRNAs, 5 in number, 41 associated proteins that add to this mass, 70 other proteins that act as splicing factors and help in binding the spliceosome to RNA substrate and its catalytic process. Another 13 proteins which are presumed to act at other stages in gene expression. Small RNA molecules occur in DNA, nucleus, cytoplasm and nucleolus and are accordingly called the snRNAs. Small nuclear RNAs, snORNAs, small nuclear RNAs, nucleolar RNA and scRNA, small nucleolar RNAs. They are found in nucleoprotein particles, snRNP. They are also designated as SNRPs or SCYRPs, SCYRPs. SNORNPs are required in processing of ribosomal RNA. Structure of spliceosome showing 6 ribonucleoprotein that are assembled at the splice site of intron or exon boundaries. The 5 snRNAs present in the spliceosome and associated with the splicing are U1, U2, U5, and U4 and U6. They are named according to the snRNAs. Each snRNP contains a single snRNA and several proteins. The U4 and U6 snRNPs are found in a single particle. Binding of U1 snRNP to 5' splice site of intron is the first step in splicing. U2 plus auxiliary factor binds to the region between the branch site and the 3' splice site. It has 2 subunits, large and small subunits. The complex thus formed is called E complex. Splice factor SF1 to BPP connects U2AF to U1 snRNP and form complex A. Binding of U5 and U4 U6 snRNPs to this complex converted into B1 complex or B1 spliceosome. Release of U1 snRNP allows U6 snRNP to interact with the 5' splice site. By this B1 spliceosome, it is converted to B2 spliceosome. Then, dissociation of UE from U6 snRNP follows U6 snRNP to pair with U2 snRNP and to form catalytic active site. U2-U6 interaction results in the removal of intron segment. Then this brings 3' end of one exon close to 5' end of the other exon, both of which join together. Steps in splicing or intron removal. Splicing of intron removal from pro-RNA involve a series of well-defined steps. A cut is made in the pre-mRNA in the 5' splice site, separating the left exon and the right intron exon molecule. The left exon takes the form of linear molecule. The right intron exon molecule forms a Lariel by bringing its 5' and 3' splice sites together. The G' base at 5' terminus of the intron become linked to base within the intron within base in the sequence that is called a branch site. A cut and the 3' splice site of intron release the complete intron in the lariat form. The 5' end of right exon is now ligated or spliced to 3' end of left exon. Lariat is debranched and the excised intron become linear. It is rapidly degraded. The 5' end of exon 1 is free to attach another intron exon junction. The branch site helps in identifying 3' splice site. It lies 18 to 40 nucleotides upstream of the 3' splice site. In eukaryotes, additional cryptic sites are present. In case, branch site is lost due to mutation. A cystic site helps in splicing. The yeast cells in the consensus sequence of branch site is given. It is highly conserved. The removal of introns from eukaryotic pre-mRNA occurs in the nucleus when the complex is called spliceosomes. Self-splicing RNA introns. Certain genes have self-splicing RNA introns. The primary transcript such as gene carries out RNA splicing on the absence of snRNPs. The intron RNA catalysis and process of splicing. Thus, catalytic RNA molecules are also called ribozymes. The process of splicing involves transesterification reaction. And RNA synthesis in vitro. 2 years before Kornberg isolation of RNA polymerase, Grunberg and some other scientists isolated an enzyme specifically involved in RNA metabolism. This enzyme was named RNA polymerase. It can link different molecules or nucleotides into long RNA chain or can break the chain into smaller sections. In the presence of this enzyme, RNA synthesis can occur even in the absence of primer in any proportion of nucleotide and even in the presence of only one kind of nucleotide. This means RNA chain can be obtained consisting solely of a single repeating base, poly A formed of adenine ribonucleotides of poly U, isolated under RNA polymerase, which could work only in the presence of RNA, a template for RNA in vitro synthesis. Substance needed for the synthesis are ribonucleoside diphosphate, adenosine diphosphate, uridine diphosphate, guanine diphosphate, and cytosine diphosphate. But all the 4 substrates need not to be present at the same time. In comparison between replication and transcription. Replication, it occurs in the S phase of a cell cycle. It's catalyzed by RNA polymerase enzyme, deoxyribonucleoside triphosphates, DATP, DGTP, DCTP, and DTP serve as raw materials. Then replication occurs along both the strands of DNA. It involves unwinding and splitting of the entire DNA molecule. It involves copying of the entire genome. Replicated DNA strand contains hydrogen bounded to its template DNA strand. 2 template strand DNA molecules are formed from one DNA molecule. Products remain within nucleus. Products are not degraded. Serves to conserve the genome of the next generation of cells and individuals. It requires RNA primer to start replication. It produces normal DNA molecules that do not need any processing. Then transcription. It occurs in the G1 and G2 phases of cell cycle. It is catalyzed by RNA polymerase enzyme. Ribonucleoside triphosphate, ATP, GTP serve as raw materials. It takes place along one strand of DNA. It involves unwinding and splitting of only those genes which are to be transcribed. It involves copying of certain individual genes only. Transcribed RNA strand separates from its DNA template strand. A single one-stranded DNA molecule is termed from a segment of one DNA strand. Greater part of the products passed from the nucleus into the cytoplasm. Products are degraded after their function is over. Serves to form RNA copies of individual genes for immediate use in protein synthesis. No primer is required to start. It produces primary RNA transcript molecules which add processing to acquired final form and size.